So let's go ahead and start with our first clicker question then. And I'll go ahead and start this up. So this is a really exciting time to be a student, I think, because instructors are trying out new things in the classroom, including completely different new formats in instruction. And so I know that the course before this one, Bio93, or its equivalent elsewhere, could have been in a number of different formats. And so I'm interested in hearing which format you actually had. So please pick which one you had for your Bio93 experience. Did you have a course that was primarily lecture? Did you have a course that was actually online? Did you have one that was flipped, meaning that the lectures were only provided online and then the lecture time was reserved for different kinds of activities? Both C and D are flipped in that sense, and I want to know whether or not you had a textbook or not. Or maybe it wasn't one of these categories, and it was some hybrid or some combination of these, in which case you would pick E. So anyway, again, if you're not getting the little green check on your clicker 2, make sure you're on the AA frequency. One of the reasons that they set frequencies is if rooms are too close together, if they're both on AA, then when you click, you end up in somebody else's data set in the same. So rooms that are close by are set with different frequencies, but AA is the usual default. And it looks like I have a question?
How do you change it?
How do you change the,
AA.
I'm sorry?
How do you change it to AA?
On your clicker?
Yeah.
Let's see, I'm not sure if I know this offhand. Let's see. Is somebody saying hold the pound sign? Hold the, oh, hold the power. Okay, thank you.
Oh, there you go. Thank you.
Awesome. Okay. Any other clicker questions? Everybody on AA? Everybody got the little green check? Fantastic. Okay, let's see what you guys picked. Okay, everybody should have clicked it. All right, let's see what you guys say. And don't put them away. We're about to have to deal with more questions. Okay, interesting. Okay, so the vast majority of you, 80%, right, if we add those two up, were either primarily lecture. So almost half of you are coming out of a primarily lecture format, and then another third a hybrid. Okay, just a tiny number of online, and then a small number of flipped. Interesting. Okay, so we'll have a small sample size for the next questions, but I'm still interested. So I'll go ahead and do these as another clicker question. And that is here. Okay, and you can actually answer this question whether you had a flipped course or not, as you'll see. So I'm interested also in not only what kind of experience or format have you had, but what your opinion or feeling is about that. So if you had a flipped course, whether it was Bio93 or its comparable equivalent elsewhere, I want to know if you actually preferred that to another format with an in-person lecture. So if you had a flipped one, then you would pick either A or B. Yes, if you preferred the flipped course, or no, you did not prefer the flipped course, but either when you had a flipped course. You would pick C or D if you actually did not have a flipped course, and then you would pick C or D depending on what your feeling was about this, right? You pick C if you did not have a flipped course and you're glad because you would prefer not to have one, or D, you did not have a flipped course but you sure wish you did. Okay, so I'm interested in did you have a flipped course or not, and whether you did or not, what were your feelings about it? So if you could please go ahead and click on that, and then I'll, everybody clicked in? Okay, watching this number. Okay, I think it's stabilized. Let's see what you guys have to say. Okay, interesting. Okay. So of the, okay, interesting. Look at this pattern here. Of those of you who did have a flipped class, most of you are, in fact, happy about that, but those of you who did not have a flipped class, which would be C or D, you're glad that you didn't. So those of you who actually experienced that are happy about it, and those of you who didn't are also happy about it. So perhaps there's some sort of self-selection going on here that you made sure that you were in or not in a flipped version of the class. Or anyway, I'll be interested in talking to you guys about that. But let's go ahead and look at one more question along the same lines, even though it's going to be quite a small sample size, at least for those individuals who've had online. But I'm interested in this because, of course, the offerings of online courses is increasing over time. So let's see, let me go ahead and start it up as I'm talking here. And I want to know, if you did have an online course, did you prefer that format to an in-person version? So you would pick either A or B. Yes, you preferred the online, or no, you did not prefer the online. So presumably, if you pick B, then that was just the primary choice you had. C or D would be if you did not have the online course. I want to know, are you happy about that, or would you have actually have preferred that? As instructors would like to know, would you prefer more online offerings, or would you prefer fewer online offerings? It's helpful for us to know that. So please go ahead and click in and let us know your opinion about that. So while I'm waiting for these numbers to stabilize, so this is an example, as I mentioned before, there's going to be two basic types of clicker questions that will be graded or scored differently. Ones like this, where there was no correct answer, any answer counts equivalently. Other clicker questions, as you'll see later in today, it will be your answer does count, meaning you get more credit if you actually pick the correct answer. Okay. My receiver certainly seems slow. Alrighty, looks like we're almost there. Everybody clicked in? Okay. Let's see what you guys think. Your opinions about online courses. Okay, so it looks like a preference for in-person courses and just a tiny preference then for online. Oh, okay, thank you for getting us back to this information to us. Okay, so let's talk a little bit about the actual format and the expectations of what's going to happen on a weekly basis. That this week was obviously very unusual since I was out of town for Monday and Wednesday. We'll not be traveling again for the rest of the quarter. So normally then, we'll meet 3 times a week in here. So it says lectures, meaning the lecture time. Much of the class, in fact, will be a lecture. We will certainly have other sort of activities and interaction things that we'll be doing. But we will be meeting 3 times a week except when there's holidays. And you will also have your discussion session 1 time a week. And you do need to go to your own discussion session. This is so that we can ensure that you all have a nice smallish group experience. Otherwise, if we allowed students to self-select at any of the times, then certain more preferred times would have hundreds of students and other ones would hardly have any. So you do need to go to the discussion session in which you are enrolled. That's a requirement. So you'll also have a discussion session. You'll also need to do the reading. This is assigned reading. I was asked earlier about what page numbers. If it just says read chapter 1, this means the entire chapter 1, which is actually only 13 pages long. The chapters are actually fairly short. And there's, of course, lots of large, attractive pictures. So there's not a ton of text for you guys to plow through. You will have on the order of about 30 or 40 pages a week. Some weeks will obviously be less when there is a holiday and so on. And in addition, you will have these assignments through Mastering Biology, between 1 and 3 of those a week. And of course, you already have had 2 that were due. So what is the relationship between these 4 different activities? Well, they're intended to be mutually reinforcing. So what I mean by that is that after a lecture, there will not be an assumption that you completely understood and processed absolutely everything. We can just sort of move on. Ditto for the reading. I will not expect that because you just do the reading that you understand it perfectly, and we can use that as a starting place. You will notice that during lecture, I often refer to the material in the reading or complementary material to that. So the idea is that by covering related concepts or examples or further discussion, that everything sort of reinforces the material so that you really thoroughly learn it. After all, that is the whole point here. So, and of course, in addition, there'll be optional office hours. So in addition to the office hours held by the tutors, I'll have weekly office hours. They're not posted yet because I'm still waiting to hear about the confirmation on the room assignment. Every single TA will also have office hours. So you're going to have a lot of different office hours to choose from. So hopefully they'll be convenient for you. Those are completely open-ended. You can show up with whatever questions you have, or you could show up just to see what questions other people have. That's absolutely fine too. Those are free form. There's no structured activity. Unlike the discussion sessions, which are not office hours, when you show up, your teaching assistant will have specific tasks for you. So, mastering biology, I mentioned that. I certainly had a lot of emails about mastering biology. I think many of the problems that some of you had with getting on the website was that you were going to the incorrect URL. Perhaps you were going to one that you had used before with a different class. There are different ways of getting to mastering biology, so you really have to use the exact URL that is provided for this course. I've also had a lot of questions about the grading settings for the quarter. If you ever want to know the grading for an assignment, you click on the blue text that's underlined that says grading policy. So for example, this is a partial screenshot from Mastering Biology for the second assignment that was associated with Chapter 1 that was due last night. And so if you wanted to know, well, how has it been graded? If you clicked on this, this blue window would pop open and show you this. So for example, the first point here is the number of answer attempts. Let's see, is 3. I don't know if you can see this. There's a big 3, right? So students interpreted this as, oh, okay, I can do 3 guesses, which is great, right? If you have a question and then there's 3 choices, it's like, oh, I am so going to get this one right, right? And then when you look at your score, you're a little unhappy to realize that, in fact, there is a penalty for picking incorrect answers, and that's explained down here at the bottom. So I do encourage you to read the whole thing down. So notice that you actually do lose points per incorrect answer. So you can guess again, but meanwhile, you have been dinged for guessing an incorrect answer the first time, but you can try again. Let's see, what else? What do you lose credit for incorrectly answering something? Hint, that is actually wrong. There must be a little bug in the window that pops up. I've set all of the grading, at least that's what it looks like on my part of the interface. The hints in no way influence your grade. If you look at them, if you don't look at them, if you answer them correctly, if you answer them incorrectly, it has absolutely no effect, or should have absolutely no effect on your score or whatsoever. If it doesn't seem to be grading like that, let me know, because that means there's a bug somewhere. But hints, you're welcome to look at them or not. It seems unfair to sort of ding you with that. You will also have a penalty for late submissions. So there are specific due dates. I think this is to your advantage, because otherwise I think it's too easy to just kind of put them off until, it's like, oh no, the test is tomorrow. I really should do all these assignments, and I have 20 of them or whatever. So the due date is to encourage you to keep up in a timely manner and go ahead and answer. These are fairly short, but they'll help give you an opportunity to self-assess if you really understand the material or not, or a little practice answering questions. And so if this time comes and goes, that doesn't mean that, bam, you've lost your opportunity. You can, in fact, go ahead and submit. You will lose 10% for each day late. So if you wake up the next morning and think, man, I forgot to submit my homework, you can go ahead and do it, and you'll get 90% of what you would have had otherwise. Okay, so you will still have an opportunity to submit. Let's see, what else was I going to say? Oh yes, the first assignment is different. This is what I'm showing you here is the default for all assignments except the first one, because the first one was sort of getting to know mastering bio. Then you will have credit for answering anything, whether it's correct or not. So I think the settings may have been wrong here. I think they've been fixed now. But anyway, I'll check them over the weekend and make sure they're all operating properly. There was enough issues with the first assignment, so I'm going to go ahead and get rid of the late penalty for the first 3 assignments, meaning the ones that were due Tuesday, Thursday, and also that are due tomorrow. So I'm going to go ahead and just remove that. So if you want to turn those in late, or if you did turn them in late, you will not be dinged for that. That in no way affects the grading of whether or not questions were answered correctly. That's completely separate, but I'll get rid of that for the first 3. So those of you who had problems getting on can be a little relieved about that. Okay, I think, okay, just one more logistical issue here, and that is that instead of using the EEE message board this quarter, we'll be using an optional or a non-UCI product, and that's Piazza. Maybe some of you have used this in other courses. It's much easier to use than EEE message board, which is why we're using it this quarter. I've been thrilled with how easy it is to use. This is completely optional. If you don't want to post questions on there, that's fine. But if you email me a question, I won't be able to deal with it because of trying to email back and forth with 800 students. But I'm certainly checking Piazza, as are all of the graduate teaching assistants and all of your peers. And so I really encourage you to participate in this, because your peers are asking great questions or making helpful comments. So this is what it looks like, at least my interface. Yours might look a teeny bit different. And so it's organized into folders by material that corresponds to different chapters. There's also one for logistics, which is what it means. It's like how the course is run as opposed to the actual content. And notice that in this one here, this particular point was, was a point about the time apparently being linked to the eastern coast, the East Coast time zone of the United States instead of the Pacific Coast, which is certainly not very convenient. Fortunately, it's easy to go and fix that, so that'll be fixed over the weekend. So if it thought you were late and you really weren't, no worries. Well, it doesn't matter anyway, because I'm getting rid of the late thing. But we'll make sure it at least knows what time zone we're in so that things are graded correctly from now on. But anyway, notice that somebody, either me or one of the graduate teaching assistants, flagged this as an instructor thinks this is a good idea. So I grabbed that and made it a little bigger so you can see that. And instructor thinks this is a good note, or good note, excuse me, not an idea, a good note. Here's a carrot for you. If you get tagged like this, then this is can either be up to 1 extra point in the clicker part of your grade, or an extra 10 points in the Mastering Biology part of your grade during the quarter. So for clickers, we're throwing out the lowest 3 scores, and so if you get a good note here, that's kind of like throwing out 4, right? So, but it won't be extra credit in the sense that the total in the clickers will never be more than 100% of the total. So if you have 100% on the clicker part of your grade, then getting lots of good notes won't actually have any significance whatsoever on your scores if you already have 100%. You won't get more than that. So anyway, but there's a little carrot for you to encourage you to point out useful things or to answer other students' questions particularly well. Okay? So let's now jump into content, and we're going to pick up a couple of points about a couple of interesting applications of evolution. So let's first consider evolution of resistance. This is a really important issue, whether we're talking about evolution of resistance to antibiotics with serious microbes, evolution of pests to insecticides or herbicides, depending on what kind of pest it is. So this is a huge issue, and there's a very nice discussion in your book about a particularly interesting case of the evolution of resistance of tuberculosis bacteria to antibiotics. So I'll just say a couple of things here. So let's consider how this can actually come about. So in this diagram, which is in front of you right now, this is a somewhat simplified version of figure 2513 from your textbook. In this particular diagram, each oval represents a bacterium. So this is different from earlier figures where ovals represented alleles. These are now just representing bacteria. And so here we have a group of bacteria. Mycobacterium tuberculosis is the species. And by the way, how would you know that this is the species? Right? You have learned that the convention that was instituted by Linnaeus hundreds of years ago is to have a two-word designation for a species. The first word is the genus, the more inclusive taxonomic category, and the second word then describes which specific species within the genus, right? So the species name as a whole is Mycobacterium tuberculosis, the bacterium that causes the terrible disease tuberculosis. So, if we consider what the bacterial population looks like diagrammatically, then in this particular case, we have most of the bacteria that are of a type that is susceptible to the antibiotic under question. And we've got a couple of bacteria that actually are resistant to the antibiotic. And if this is in fact the case in the population, then what this means is that if we add the antibiotic to the patient's body, which is the environment for these tuberculosis bacteria, then what happens? The ones that are susceptible are gone, right? If they're killed off by the antibiotic, what's left? The bacteria that are actually resistant to the antibiotic, which means that they can then go ahead and proliferate even in an environment where there is antibiotic. And so the antibiotic-resistant bacteria then can grow and spread. And of course, this is a terrible problem. And in the case that was discussed in your book, the patient, in fact, eventually died, even though the extraordinary facts associated with this case is that it was a single point mutation in the bacteria that allowed them to that conferred resistance to antibiotics in these particular bacteria. So it can be a relatively small change. It doesn't have to be, but it was in this particular case. Now, one really important thing to note here that students often miss is that the antibiotic did not cause the mutation, right? The mutation was already there. Let's go back in time before we even added the antibiotic. Here we are. The bacteria with the mutation were already there. The antibiotic is not causing the mutation. What the antibiotic is causing is that the bacteria that are not resistant to the antibiotic are now gone, leaving the other bacteria to proliferate. So all the antibiotic does is just causes differential mortality or selection then on these bacteria. So we end up with a different bacteria with a different genetic makeup after the application of antibiotics. Now, of course, this is really overly simple, as you can imagine. Right, we treated this as if it was purely dichotomous, right? They're either completely susceptible or not. And so, in fact, the true case is usually a bit more nuanced. So, for example, I can indicate that by making some bacteria which sort of have some resistance, maybe not complete resistance, but some, right? And so in this kind of case where you have a population of bacteria that differ in their susceptibility, it should make sense to you that under these conditions then, if a patient doesn't complete their course of antibiotics and doesn't completely wipe out all of the bacteria that have any susceptibility at all, then the bacteria that do have some resistance at some level, then if they're not killed off, upon further replication, then they can accumulate other changes and end up resistant to the antibiotics. So that's why one of the major points that's often made in public health is make sure that patients take their full course of the antibiotic rather than just partially killing off the most susceptible and leaving pathogens that have just some fractional amount of resistance. So tuberculosis may be less familiar to some of you because, in fact, in North America, it's not as big a deal as it is elsewhere in the States. This is not a graph from your book. This is, I believe, from the CDC, the Center for Disease Control, and this particular graph then shows the incidence of tuberculosis as of 2007. So these numbers here on this color scale show the number of tuberculosis infections per 100,000 people. And so the blue areas have far fewer cases, which includes all of North America, parts of Europe, and also Australia. You can see the parts of the world that are, the more yellow it is, the more incidence of tuberculosis there are, and you can see that Africa is particularly impacted, and parts of Asia and Russia as well. So this is tuberculosis as a whole, not necessarily tuberculosis strains that are resistant to antibiotics. But this is extremely extensive, and tuberculosis is a frightening disease because not only does it have a very high fatality rate if it's not treated with antibiotics, but the course of antibiotics goes on for months. That is a detail that you may have missed on your reading of the case in the chapter. But typically months of taking antibiotics is required in order to get rid of the bacteria in a patient's system. And although with the advent of the antibiotics in the early 20th century, the tuberculosis impact globally declined tremendously, it then came back in the 1980s. Why? Because of spreading resistance to antibiotics that were in fact readily available via mechanisms that are very comparable to the one that you read about in the book that I just mentioned. And so by 1993, the World Health Organization, in fact, declared it to be a global health emergency again because of the spread of antibiotic-resistant tuberculosis. So this is still considered a really terrible threat, even though here in North America it is less directly impactful. But at a global scale, about 1/3 of the entire population in the world either has or has had tuberculosis. So this is a really terrible problem. I'm going to relate this to something which might seem unrelated, and that is the process of pasteurization. And I was rather astonished to learn from some other students that in fact they were quite familiar with the idea that pasteurization, a process to which some foods, particularly dairy products, are shown. So here's a picture of some cartons of milk, and notice that whether it's low fat or fat-free or whole milk, all of these different kinds are in fact shown as pasteurized. This verb relates back to in honor of Louis Pasteur, the rather extraordinary French scientist who did those amazing experiments with the flasks to test the idea of cell theory, that cells in fact come from pre-existing cells and are not coming from some form of spontaneous generation. The same Pasteur. Apparently that's, students are fairly familiar with the idea that this in fact relates to Louis Pasteur. But how does this relate to tuberculosis? How does tuberculosis spread? It's a respiratory disease, and so it's extremely contagious. If you're talking to somebody, they have tuberculosis and they cough, you can catch it, okay, the airborne bacteria. So it's extremely contagious compared to many other serious diseases. If we go take a little field trip here to the Center for Disease Control, which is one of the most authoritative, best websites in the United States for information on various diseases, we're visiting right now a page that says food safety and raw milk. Why are we going to raw milk? Well, I thought this was interesting and I would share it with you. This starts by saying back to nature. That's what many Americans are trying to do with foods they buy and eat. They're shopping at farmers markets, picking organic foods, participating in food cooperatives, growing their own food, trying to eat foods with minimal processing. And so this is certainly a tremendous cultural change in this country as people try to eat and consume in a more healthy manner. But this also includes consumption then of raw milk. And the Center for Disease Control, which usually doesn't involve itself in such issues unless there are health issues associated with these, goes on to point out that milk and products made from milk are foods that when consumed raw can pose severe health threats. Milk products need minimal processing called pasteurization, can be done by heating the milk briefly. Those of you who may not know the details might be astonished to see that it actually can be done in a few seconds. For example, heating to 161 degrees Fahrenheit for about 20 seconds. So interestingly, it's actually different from sterilization. Milk is not sterile, it's not sterilized, it's just treated briefly with enough heat to kill the usual disease-causing germs. Now, what does this have to do with tuberculosis? You know, did somebody like cough on the milk before it was packaged? You know, what's going on here? Interestingly, okay, look at this first sentence here. Before the invention and acceptance of pasteurization, raw milk was a common source of bacteria that cause tuberculosis. Tuberculosis is the first disease listed because that was one of the major sources of tuberculosis before pasteurization, and that should strike you as kind of odd. The reason for that is that cattle also are infected with the same bacterium, and this is something that cause tuberculosis in humans as well. So we can get tuberculosis from the bacteria on cows that then enters the milk during milking. And this is why the pasteurization of milk then protects us from tuberculosis if in fact the cows had the same bacterial species. So there are a variety of other kinds of diseases as well, but I thought you would find that interesting to link back to Pasteur. Anyway, let's now consider a slightly different topic. So I was thinking of evolution of pathogens. Let's now consider a curious case of evolution of hosts and their parasites. And although we'll be discussing a specific case, many of the things that we say, of course, are general concepts, just as what was discussed with evolution of resistance to antibiotics versus susceptibility in the tuberculosis bacterium, tuberculosis-causing bacterium. Many of those concepts can be generalized across different kinds of pathogens. So what are we going to consider? We're going to consider some unsavory ectoparasites, namely lice. Okay, so these are parasites on the outside of bodies, and they're really quite common on a variety of different kinds of mammals. And lice are insects. Here are some pictures of some lice in front of you right now. Lice are interesting because they specialize on a single host species. And so the lice that you find on one particular species of mammals, for example, you would not tend to find on a different species of mammal. And so if one looks at the primates, then humans are unusual in that we have not one, not two, but three completely different types of lice potentially on our bodies, although our primate relatives only have one louse species associated with them. So what are these three types of lice anyway? Here they are. On the left, we've got the so-called head louse. This is a louse that, by the way, lice is plural, louse is singular, right? The head lice live then on the scalp or in the hair of the head. The second louse here, the pubic louse, lives in pubic hair, so associated with external genitalia in our species. And then the third charmer here on the right is the body louse. And unlike the first two, which live on the skin and the hair, the body louse primarily lives on clothing. And so all of them are blood-sucking, so the body louse then leaves the clothing to feed on the human skin and then goes back and hangs out in the clothing. So head lice are then spread between individuals by contact with contaminated products like combs or brushes or scarves, or you can even touch somebody's head or put your head on a headrest somewhere where somebody with head lice was. I know everybody's getting a little itchy, aren't you? Pubic lice usually are spread through sexual encounters. The body louse on the far right, though, can be spread by contamination with contaminated clothing. So what does this have to do with evolution of lice or hosts and their parasites? Before I leave this slide, I also want to point out the species here. Here they are. Notice there's two different genera, right? These two are the same genus, right? In fact, they're even the same species, right? Pediculus humanus. So what's that third word? That third word there, capitis or corporus, indicate that there are varieties or subspecies of this same species of louse. They're sufficiently distinct that they're actually given names, and there are many differences between them, but at this point in time, they are still considered a single species. But they're quite distinct from the pubic louse, which is not only a different species, but it's a completely different genus. So if you were going to draw a phylogenetic tree of these, then what would the relationships of these three be? Right? You can tell just from the taxonomic names, right? These two here would be sister species, right? So that'd be pediculus, right? Pediculus. And this one would be over here, right? Does that make sense to everybody? These two are more closely related to each other than either one is to this one because they're grouped, and you can tell that just straight from the taxonomic name. Okay, so now if we leave the lice for a second and their relationships and move on to some primates, then of course The relationship between, the taxonomic relationship between different primates is fairly well known. This is just a small handful. Here we are, our closest group, at least on this particular tree, are the chimps, and so we share a more recent common ancestor, which of course would be right here on this phylogenetic representation. Notice that these have angled branches. This is no different from drawing something with more right angles like this, right? In your book, they tend to look more like this, or even like this sideways. It's the same information, whether it is angled or at right angles or pointing up or pointing down. You should still be comfortable with the idea that time in this particular case is going in this direction, right? Meaning that going down in that direction is going earlier and earlier historically, right? And in this particular one, time is going like this, and in this one, time is going like that, right? You should be very comfortable flip-flopping back and forth between these different kinds of representation. Okay, and then the other primates here are shown as less closely related to humans than chimps. Okay, so it is thought that humans and chimps diverged from each other about on the order of maybe 6 million years ago or so. So if we look at this point in time, then that would be this point right here. So about 6 million years before present, right? Looks like it's mind your own beeswax or something, but this actually stands for million years before present, okay? Our particular species is thought to have evolved about 200,000 years ago, and there were several other species of so-called hominins. You might say, what is a hominin? And that is defined right here in the third bullet point. It's any species that is more closely related to a human than to a chimpanzee. So what does that mean? If we go back to this tree, then that would mean anything that would be branching off here and went extinct or so on. It is more closely related to a human than to a chimp. Okay? So we do know some times for some of these divergences in the lineages. Okay. So there might be some questions about hominins that the fossil record can't really answer, which are things like, when did hominins lose their body hair? So for example, we have far less body hair than our other primate relatives. And so it's of interest to think, well, when do we lose these? And also, when do we start wearing clothing? After all, the other primates don't wear clothing. And you see where we're going with the whole lice issue here too, right? There are various ways that you can think to address this question. You can look for historical evidence of tools to make clothing. They've actually found needles that are 40,000 years old that they think were needles made to use clothing. So this seems to be fairly ancient. And you can also look at past climate maps and think about how this might relate to hair distribution or clothing and so on. But it's not completely direct evidence, but maybe we can use divergence in lice to figure this out, right? Can we look at lice, the evolution of lice? And in fact, scientists have done this, analysis of evolution in lice to infer when we and our relatives lost our body hair in evolutionary time and started wearing clothing. So let's try one to see how this works logically. Human head lice and chimp lice, and notice it doesn't say chimp head lice, right? Because unlike humans, all the other primates have just one species. So chimp lice are then all over a chimp's body. Okay, so human head lice and chimp lice belong to the same genus. Why do you think these two lineages actually diverged? We're running out of time here, so normally I would have you work with a neighbor and consider hypotheses, but I'm just going to go ahead and charge forward without giving you time to work with your neighbor, because I want to do this as a clicker question in just a second here. So you might consider, for example, that there was some co-speciation going on, such that when humans and chimps diverged about 6 million years ago, right? If when humans and chimps diverged, this was also a time when lice on humans, at least on human heads, diverged from chimp lice, then one would expect it to be at about the same time, right? And how would you test this? Well, you can do this by comparing the DNA of head lice to DNA of chimp lice. And if you use then molecular clock reasoning, which I'll go through on Monday, to estimate when they actually last shared a common ancestor, you can try and put dates on this. So to see if you understand this, I'm going to do this as a clicker question. And notice this is an example where the answer counts, meaning you get more credit if you answer correctly than incorrectly. So I'll go ahead and walk through it with you. So what would you predict if we assume the co-speciation hypothesis is in fact true, that what's driving the divergence of the lice is the divergence of their hosts? So, given the relationship between humans and all of the various other primates that are shown on here, when would you assume then, if this is true, when would the last or most recent common ancestor of chimp lice and human head lice, so I wrote lice in all capitals, so we're talking about lice here, not the primates, but the lice themselves, when would the last common ancestor live? 20 million years ago, at this point on the graph, right? 6 million years ago? Or 100,000 years ago? If you're not sure about the logic, this would be a great time to chat with your neighbor, okay? Be friendly. If you don't know them, hello, my name is, I have no clue, what do you think? Or, well, this is obviously the answer. I'll give you 30 seconds or so and then check in with you. Okay, everybody clicked in? If you click in more than once, by the way, we'll just use your final answer. Okay, everybody clicked in? I'll show you what you guys picked. Everybody in? Yikes. Okay, looks like that's the right number here. Let's see. Okay, woo! Awesome! Great job, everybody. Hey, go have a great weekend, and I'll see you on Monday.